in this line of work. Before I get into everything, I do just want to take a moment to recognize my postdoc mentor, Dr. Monique LeBourgeois, who unfortunately passed away last fall. She pioneered much of the work that I'm going to be presenting today, and she developed all the methods that we used for conducting circadian research in early childhood. And she told me that when she first came up with this idea to do these rigorous circadian protocols with young children, she was told that she was crazy and it was impossible. But as you'll see, she proved that it can be done. She also knew that it was critical to expand our knowledge of circadian physiology in early childhood, which is a sensitive period in the development of sleep and circadian regulation, and a time when sleep problems commonly first emerge. And so, in fact, approximately 25% of young children experience some form of behavioral sleep problem, including difficulty falling asleep, bedtime resistance, or nighttime awakenings. These problems often persist throughout the school age years and are associated with both concurrent and later attentional, emotional, and behavioral problems, as well as increase in cardiometabolic risks. But there's still a big gap in our understanding of the mechanisms that underlie the development and maintenance of these early behavioral sleep problems. And so my research focuses specifically on how the lighting environment contributes to young children's circadian physiology and sleep health. This is likely review for a lot of you, but I just wanted to take a quick step back and look at the mechanisms by which light influences sleep and circadian timing. And this occurs primarily through stimulation of the eye's intrinsically photosensitive retinal ganglion cells, or iPRGCs, by light. And these are photoreceptors in the eye that are maximally sensitive to short wavelength light, with a peak sensitivity to blue light at approximately 480 nanometers, and they express a photopigment called melanopsin. So when stimulated by light, the signal is transmitted from the iPRGC to the suprachiasmatic nucleus in the brain, which is the central circadian clock, and regulates rhythms throughout the rest of the body. The SEN then modulates production of the sleep-promoting hormone melatonin in the pineal gland, and exposure to light at night can suppress melatonin and delay the timing of the circadian clock, whereas exposure to light in the morning advances circadian timing. So research is still very scarce on the development of the human iPRGC, but what we do know suggests that the system emerges early in an individual's development. So that photopigment melanopsin has been observed in human eye tissue as early as 8 weeks post-conception. And signals from the iPRGC also contribute to the pupillary light reflex, or PLR, which controls the diameter of the pupil in response to a light stimulus. So we can also look at the development of the pupillary light reflex as an indicator of when those iPRGC come online and become functional. And so this figure on the left depicts the number of premature babies at each week of gestational age who display a pupillary light response, which are the shaded bars, or those who don't, which were the blank bars. And so as you can see, the pupillary light response emerges in preterm infants between 30 to 35 weeks gestational age, again suggesting that the human iPRGC are sensitive to light input by full-term birth in humans. Additionally, this study examined the PLR in response to blue and red light at 2 different intensities in premature infants at 38 weeks gestational age. And in all of the babies that they tested, the red light did not induce pupil constrictions for either intensity. However, the pupillary reflex was elicited by the blue light with a stronger response to those brighter intensities, suggesting that the early PLR in human infants is predominantly mediated by the iPRGC, even at 38 weeks gestational age. Lastly, several studies have examined the effects of cycled lighting, meaning lights on during the daytime and then lights either very dim or completely off during the nighttime on preterm infants in the neonatal intensive care unit. And data from these studies indicates that exposing premature infants to a light-dark cycle rather than a constant light environment is associated with earlier distinct patterns of rest and activity, a better growth rate, and a shorter hospital stay. So taken together, all of these studies provide evidence of light detection and entrainment early in human development, and also highlight the importance of environmental signals to the development of the clock and for sleep. So jumping ahead from infancy, we know that an individual's circadian sensitivity to light changes throughout the lifespan. Data from several studies actually suggest that children are more sensitive to light exposure than adults. In this study depicted here, this is 9-year-old children who showed almost twice as much suppression to an evening bright light exposure compared with their parents. Similarly, previous work from Dr. Stephanie Crowley demonstrated that pre to mid-pubertal adolescents have significantly greater melatonin suppression than late to post-pubertal adolescents in response to evening light exposures across 3 different intensities, suggesting that there's actually a reduction in light sensitivity even across child development. There's still little research on photosensitivity in early childhood. In this study by Dr. LeBourgeois, 21 preschool-age children followed a stable sleep schedule and wore a pendant light meter for 4 days. Then they completed an assessment of their dim light melatonin onset, or DILMO. And they found that when controlling for children's bedtimes, those who were exposed to more light in the 2 hours before bed had later DILMOs. And so this tells us that the evening lighting environment does play a significant role in the timing of the circadian clock in this early age group. So the main focus of my research over the last few years has been on how the characteristics of light affect the circadian clock in early childhood. And today I'm going to present data from 3 different studies examining some of these characteristics, one of which is already published, and the other 2 are new studies that are unpublished. So in our initial study, we sought to establish dose response curves to evening light. To do this, we examined the impact of exposure to different intensities of light 1 hour prior to children's habitual bedtimes on their melatonin levels and circadian timing, and this is in preschool-age children. So just some quick information on the units that I'll be using throughout the rest of the presentation to make sure we're all on the same page. Intensity can be measured in a variety of units, but the work I'll be discussing, it will be primarily discussed in terms of illuminance, the unit of which is lux. So for anyone not familiar, 1 lux is defined as the brightness of 1 candle, 1 meter from the eye. And so moonlight is very dim at about 0.1 lux. Daylight is very bright at 10,000 lux or greater. But important values to keep in mind are that typical room lighting is usually somewhere between 100 to 300 lux, and an iPad screen at full brightness 1 foot away from the face is about 100 lux. And so these are common sources of light that children might be exposed to in their everyday environment in the evening. The spectrum of white light can be described in terms of its correlated color temperature, or CCT, the unit of which is Kelvin. And higher CCT values describe a cooler light that typically contains more blue light, and lower values describe more of a warm light that typically contains less blue light, more red light. So here are examples in this image of a lower CCT light source on the left and a higher CCT light source on the right. But circadian researchers have moved away from talking about CCT because it's not really an accurate representation of the spectrum in all cases. So 2 very different light spectrum that will have different impacts on the circadian system can actually produce the same CCT. And so a better way to quantify the circadian effects of light in terms of both spectrum and illuminance is with melanopic equivalent daylight illuminance, or MEDI. This newly developed official SI unit describes the influence of light on the IPRGC and strongly predicts the circadian response to light exposure. Recent recommendations for light levels to support sleep and circadian rhythms in healthy adults were published, and they suggest exposure to a minimum melanopic EDI of 250 lux throughout the day. Then they recommend that starting at least 3 hours before bedtime, individuals should be exposed to melanopic EDIs of no greater than 10 lux as measured from the eye. Remember that this is recommendations for adults, and as we'll see in the rest of my data, this may not necessarily be true for children, and we might need different recommendations to support children's sleep. But back to light intensity and our first study trying to establish illuminance response curves. So in adults, we know that even low levels of light can affect circadian rhythms. Furthermore, as you can see in these figures, light exposure in the evening both suppresses melatonin and delays circadian timing in a dose-dependent manner with intensity. So at low light intensities, there's not really much of a response, but that it gradually increases with brighter intensities up to a saturation point with very bright intensities. So in these data, after a 6.5-hour nighttime light exposure, 50% of the maximal phase shifting response was observed after exposure to light of about 100 lux. So that's well within that range of typical indoor room light. A more recent paper has demonstrated that the adult circadian system may be even more sensitive to light than was previously thought. So here, participants were exposed to 5 hours of light in the evening across a range of light levels between 10 to 2000 lux. And across the entire group, they found that the 50% melatonin suppression response occurred at an average of only about 25 lux. So for our study, we collected data from 33 children ages 3 and 4 years. All participants in this and all the studies I'm going to present today underwent a rigorous screening process to ensure that we only enrolled healthy children with no sleep, circadian, or ophthalmological disorders. And our main measure throughout was salivary melatonin, which we collect with young children by having them chew on a braided cotton roll for about 2 minutes, as you can see here in this picture, until the cotton is saturated with saliva, which is then placed in a tube and immediately centrifuged. This figure depicts our study protocol, which lasted for a total of 10 days. For the first 7 days, parents keep their child on a strict sleep schedule, and the child wears an actigraph and a pendant light meter throughout the study, and the parent completes a daily sleep diary. Then the final 3 days of the study consist of a dim light circadian assessment, which we call cave days with the children, and we use child-friendly language throughout all these kinds of protocols to make sure that the science feels less clinical and a little bit more fun and approachable for them. And as you can imagine, it might be difficult to get preschoolers and their parents to live in a sleep lab for 3 days. And so for all the studies I'm going to present today, we actually conduct them in participants' homes, where we recreate well-controlled laboratory-like conditions. So here's an example of our team in the process of setting up a home for dim light. The first thing we do is we cover every single window in the home with black tarp, and then we ensure that all light levels in the home are less than 10 lux of the child's angle of gaze. We do this by covering, dimming, or redirecting home light fixtures, or setting up our own dimmable lamps at strategic locations. We then have to address home appliances that light up, or for children, their toys, their shoes, their toothbrushes, all the things in the house that might have a light on them. We have to find and test and ensure that they're below 10 lux at anywhere the child can get to, or we disable it or remove it for the duration of the in-home assessment. And so throughout the duration of the dim light assessments in this first study, children were exposed to an average of only about 1.5 lux as measured from their wrist. So on day 8, children enter the dim light environment 4.5 hours before their scheduled bedtime. We then obtain a baseline assessment of their DILMO by collecting saliva samples in 20 or 30-minute intervals throughout the evening until 1 hour past children's habitual bedtimes. On the evening of day 9, children were exposed to light for 1 hour before the habitual bedtimes. And the way that we do that is we have them play at a dimmable LED light table in order to make sure that their gaze is consistently focused down on the light source throughout that entire hour. So we'll have them color on clear overhead transparencies or play with translucent blocks or just anything we can think of to make sure they're looking down throughout. Each child is randomly assigned to one light intensity between 5 and 5,000 lux, and the light source that we used for this study had a CCT of 5,000 Kelvin. Finally, on the 10th night, we measured their DILMO a second time, taking samples until 2.5 hours past their bedtime in order to make sure that we captured any phase delay. So the a priori hypothesis for this study was that light before bed would both suppress melatonin and induce a circadian phase delay in a nonlinear, intensity-dependent manner. So similar to what we've observed with those curves in adults, but maybe with the curve shifted to the left since we expected them to be more sensitive based on that previous work with older kids and adolescents. But instead, here's what we observed. This figure depicts percent melatonin suppression across each of the experimental light intensities. And we see that children had a very high level of melatonin suppression in response to light across the full range of intensities. Melatonin suppression ranged from 69 to 99%, with an average suppression of 85%, and no clear relationship with light intensity was observed. However, when we break the light intensities into quartiles, we do now observe that children who were exposed to the lowest quartile of light intensities, so between 5 to 40 lux, did have significantly less melatonin suppression than those who were assigned to each of the 3 higher quartiles. But keep in mind that the average suppression in that lowest quartile was still 77%. So if we rescale this figure with the y-axis now starting at 0, it's very clear visually again that children's acute melatonin response was highly sensitive even to those lower light intensities. Here we see the observed phase delay in minutes for each individual child, and negative numbers in the figure indicate a phase delay. The phase shift ranged from an 8-minute advance to a 123-minute delay. And on average, DILMO was delayed by 56 minutes on day 10 as compared to baseline on day 8. Again, we did not observe any clear relationship between light intensity and the magnitude of the phase delay. Rather, we see robust phase delays across the full range of light intensities, even at those low intensities. So together, the findings from this initial study demonstrate that young children's melatonin production and circadian timing may be highly sensitive to light exposure in the hour before bedtime across a wide range of light intensities. So with this basic information on children's sensitivity to evening light intensity, our next step was to examine how the spectrum of light impacts the developing circadian system. So a little bit of background about what we know about sensitivity to light spectrum in adults and older children. In this study with adults, they were exposed to 2 hours of an evening white light with a brightness of 40 lux. And they measured melatonin levels after exposure to 3 different CCT light conditions. Results showed significantly more melatonin suppression as a result of the higher correlated color temperature light at 6,500 Kelvin that contained more blue light compared to each of the lower CCT light conditions at 2,500 and 3,000 Kelvin. Similarly, here's a study from Dr. Higuchi's work of 9-year-old children and their parents who were exposed to a low CCT or high CCT light condition at night and the resulting melatonin suppression. Light intensity for this study was quite a bit higher than the first study at 300 lux. So that's towards the higher end of what's typical for indoor room light. And you can see that the children had significantly more suppression in response to the high CCT light than the low CCT light. So in suppression of 81% compared to 58%, whereas no difference in suppression between the two light conditions was observed in the adults, again demonstrating greater light sensitivity in children than adults. Finally, for both adults and school-age children, the average CCT of light measured in the home environment is significantly associated with DILMO timing. So those who had, on average, higher CCT lights in their home also had significantly later DILMOs, suggesting that high CCT light levels at home could contribute to a delay in children's circadian timing. So for our study, we performed a within subjects protocol to examine the sensitivity of the circadian clock to differences in light spectrum in preschoolers ages 3 through 5. We used the same 10-day protocol that I described before, with the one exception being that we shifted the timing of the light exposure 30 minutes later to now be centered at habitual bedtime. Each participant completed the protocol twice, with only the light condition that they received differing between the protocols, and the order of that was counterbalanced across participants. So we compared melatonin suppression and phase delay in response to this evening light exposure to a high and low CCT LED light. And you can see the two light conditions that we used depicted on the right, along with the melanopic EDI for each. And the light conditions were held constant at a photopic illuminance of 20 lux. The spectrum that we chose for this was in order to ensure high ecological relevance, as there are light bulbs on the market today that are commonly sold with spectra at both 2,700 and 5,000 Kelvin. And so these are light spectrum that a child could be conceivably exposed to in their everyday life in their home environment. We also decided on a photopic illuminance of 20 lux, again for ecological relevance, to approximate an iPad at about half brightness. The 5,000 Kelvin light source on the bottom was very similar in spectrum to the one that we used in our first study on light intensity. That one at 20 photopic lux had a melanopic EDI of 16.8, so very similar spectrum, similar circadian stimulation. So we would expect participants in this study would also have a high sensitivity to that condition. But we want to know if, compared to that, exposure to a warmer white light containing less blue light, presumably less stimulating to the IPRGC, would result in less melatonin suppression and phase delay in these highly sensitive young children. So here we have average melatonin curves for the baseline and light exposure nights during the 5,000 Kelvin protocol. The closed circles represent melatonin levels in the samples taken during the baseline evening, and open circles represent those taken during the light exposure night, with the timing of the light exposure highlighted in blue. So during the 5,000 Kelvin exposure, melatonin was suppressed an average of 56.3%. And here we have average curves for the 2,700 Kelvin exposure, during which melatonin was suppressed an average of only about 24%, and that's significantly lower than during the 5,000 Kelvin light. Additionally, melatonin levels remained significantly lower than the baseline night 20 minutes after the end of the 5,000 Kelvin light exposure, but not after the 2700 Kelvin exposure, indicating that children had a more sustained response to that high CCT light. Unfortunately, we were unable to compare phase shifts across light conditions for 4 of our participants due to either accidental light exposures or protocol errors. So here we have complete phase shift data across both light conditions for only 6 of the children. Here again, closed circles are saliva samples taken during the baseline night, and now here open circles represent those taken on the final night of the protocol. And so we observed significant delays across both light conditions, with an average delay of 39.6 minutes for the 5000 Kelvin condition and 26.7 minutes for the 2700 Kelvin condition. While on average, the phase delay was 13 minutes larger after the high CCT light condition, the difference in magnitude of phase delay between the 2 light conditions was not statistically significant. Importantly, this is only 6 kids, even though it's within subjects, so it's likely that our power is too limited to detect a significant difference between conditions. But you can still see that effect sizes were similarly large for both light conditions. Additionally, the pattern that we're seeing here of our findings does mirror those of a study by Dr. Hannafin and Dr. Brainerd, in which adults had greater melatonin suppression in response to a 17,000 Kelvin, very high CCT light spectrum, compared with a lower 4,000 Kelvin light at around 100 lux. But they found no significant differences in the magnitude of the phase delays between the 2 light conditions. So very similar to our pattern of findings, a significant difference in the suppression response to 2 different lights of different spectrum, but no difference in the magnitude of the phase shifts and pretty robust phase shifts in response to both conditions. So this could suggest that maybe there's differences in spectral sensitivity in that acute suppression response compared with how the light signals get integrated into the slightly longer outcome of melatonin onset the following evening. We do know that light-induced melatonin suppression and phase shift are functionally independent, and so it's possible that these outcomes are differentially sensitive to light spectrum. But to summarize the findings of this study, we observed preliminary evidence that the circadian clock in early childhood is sensitive to differences in evening light spectrum, with increased suppression of melatonin observed in response to light of a higher CCT compared to light of a lower CCT. However, both light conditions at a photopic luminance of only 20 lux resulted in significant delays of melatonin onset, adding again to a growing literature demonstrating high sensitivity of the early circadian system to evening light exposure. And an important implication of this high photosensitivity to light in the evening comes from the fact that young children generally do not get to choose their own bedtimes. Although adults typically self-select bedtimes about 2 hours on average after melatonin onset, parent-selected bedtime in toddlers and preschoolers occurs an average of only about 40 to 45 minutes after melatonin onset. And in about 15 to 20% of the data that we've collected on circadian timing in preschoolers, parent-selected bedtime actually occurs before melatonin onset. And this is important because in a previous study, Dr. LeBourgeois demonstrated with regularly napping toddlers ages 2.5 to 3, which is depicted here, that as bedtime gets closer to children's melatonin onset, so as bedtime phase of angle of entrainment gets smaller, children take longer to fall asleep and have increased bedtime resistance, likely because they're being put to bed at a time when they're just not physiologically ready to fall asleep yet. And we find that this association holds between bedtime phase angle and sleep onset latency, even for children ages 3 through 5 who are no longer napping. So we can see now in this figure that these children are being put to bed before their DOMO has even occurred, and that they also have the longest sleep onset latencies. But with such a small phase angle of entrainment between DOMO and parent-selected bedtime throughout early childhood, a light-induced phase delay of the magnitudes that we're observing in our first 2 studies here, even in response to those dimmer light intensities, would push DOMO very close to or even after parent-selected bedtime for a lot of young children. So if we had a light-induced phase delay of 30 minutes, now all of these children are also being put to bed before their DOMO. And this mismatch between their bedtime that parents are choosing and their circadian timing might be making it harder for them to fall asleep and could be contributing to the high prevalence of behavioral sleep problems that we observe at this age. And we know that young children are frequently being exposed to artificial light at night in the home, including in the form of media devices. A 2020 survey by Common Sense Media reported that nearly half of children under age 8 use screen media in the hour before bedtime. Those data were collected pre-COVID, so we also looked at some recent data that we had in a survey looking at evening media use that was collected last year. This is over 1,000 parents of children ages 1 through 13. Here in this figure, I've just focused on preschool children ages 3 through 5. So this is data from 351 preschoolers throughout the US. And we observed that 62% of them used screen media in the hour before bedtime at least once per week, with 20% of them reportedly using it every evening. And so children's frequent media use is consistently associated with negative sleep outcomes, such as later bedtimes, longer sleep onset latencies, and shorter total sleep durations. And our data suggests that this could be due in part to the circadian effects of the light exposure from the screens due to the high sensitivity in early childhood. So let's talk briefly about why young children might be so sensitive to evening light exposure. One possible reason is because of developmental changes in anatomical features of the eye, specifically that children have both larger pupils and clearer lenses than adults, which is just going to allow more light into the eye to stimulate those retinal ganglion cells. As humans age, the ocular lens becomes more yellow, making it more difficult for blue light in particular to get through. So on the left, you can see images of 6 human donor lenses at different ages, and that they become progressively more opaque with increasing age. The figure on the right depicts lens transmittance for different wavelengths of light for school-age children in red and their parents in blue. And at 480 nanometers, which remember is the peak sensitivity of the IPRGC, transmission through the lens was measured at 8.6% higher in the children than in adults. Additionally, they calculated that non-visual photoreception was 1.48 times greater in the children than in the adults. Regarding pupil size, this figure on the left, pupil diameter is compared between school-age children and their parents under both dim and bright light conditions. And under both conditions, children have significantly larger pupils than their parents. In fact, pupil diameter increases across child development, peaking around late adolescence and then slowly decreases again throughout adulthood. So these differences in eye anatomy likely contribute to children's high photosensitivity, but there may also be differences in downstream processes from the eye that have yet to be explored that contribute to this high sensitivity in early childhood. For the final study that I want to present on, we wanted to extend what we've learned so far about early photosensitivity to evening light in early childhood, and now establish the dose response curve to morning light in preschool age children, examining the phase advance response to 1 hour of morning light across different light intensities. The protocol for this study is very similar to the ones that we used for the 2 earlier studies. We have the same 7-day lead-in period with children following a strict parent-selected schedule and at-home actigraphy, followed by an in-home dim light circadian assessment. On the first evening in dim light, we didn't collect any saliva samples. The children went to bed at their normal bedtime. We just wanted them in dim light and ready to go for the next morning. And on the morning of day 9, we woke them up 1.5 hours before their habitual wake time so that we could assess the morning decrease in melatonin and calculate a baseline DIL-MOF. That evening, we then collected saliva samples to assess baseline DIL-MO. On day 10, we again woke children up early and then did a 1-hour light intensity ranging from 1.5 to 2,000 lux, starting at their habitual wake time. And we used the same 5,000 Kelvin LED light panel from the first study. Then that evening, we again collected saliva samples to assess their DIL-MO to see if there were any changes from the previous night after the light exposure. We started collecting saliva samples on that final evening 2 hours earlier than the baseline night in order to make sure that we still captured the DIL-MO if there was a large phase advance. Lastly, on the morning of day 11, we woke children up again 1.5 hours early and collected saliva samples until 3 hours after habitual wake time in order to assess any changes in DIL-MOF. This was a really tough protocol to enroll families for. Some preschoolers naturally wake up at like 6 a.m., so you can imagine that parents weren't really thrilled about signing up to have us come to their house and wake their child up at 4:30 a.m. for 3 days in a row. So in the end, we were able to get 13 participants to complete the full protocol. So here we have average melatonin levels on the baseline morning, starting 1.5 hours before children's habitual wake time. You can see that we do have an initial decrease in levels, but that we don't really see a sustained drop below the threshold of 4 picograms per milliliter. In fact, we were unable to calculate a baseline DOMOF for a number of kids because their melatonin levels never went below 4 picograms per milliliter, even 3 hours after their habitual wake time when we stopped sampling. Or they would dip below and come back up and never remain sustained below that DOMOF threshold. But at the same time, for several of the other kids, we were unable to calculate their baseline DOMOF because their melatonin levels were already below 4 picograms per milliliter when we woke them up an hour and a half before their habitual wake time, and then the levels remained low throughout the morning. We actually collected samples to assess baseline DOMOF across 20 children, and so here you can see the larger sample, and that pattern holds across even that larger sample. So we were only able to calculate a baseline DOMOF for 10 out of 20 kids because the samples were either already below our threshold or never went below our threshold. So to my knowledge, this is the first attempt to assess melatonin offset in early childhood. And so clearly we need a lot more research, in particular to measure the full curve of nighttime melatonin production in this age group, so that we can get a clearer picture of the time course of salivary melatonin in early childhood, understand the length of the biological night, and how melatonin onset and offset compare to children's bedtime and wake times. But now here we have melatonin levels in orange on the second morning of the protocol, during which children received the 1 hour light exposure, with the timing of which is highlighted in yellow. And as we would have expected, we're not seeing an acute drop in melatonin levels during the light exposure, suggesting that melatonin synthesis has already ceased at this point, and there's no acute suppression response to this morning light exposure. Finally, here are melatonin levels on the final morning of the protocol. Within individuals, we found that the first sample on that final morning was significantly lower than the first sample on each of the previous 2 mornings, possibly suggesting an advance in the timing of melatonin offset as a result of the previous day's morning light exposure. But unfortunately, because we were unable to calculate DOMOF for both the baseline and final mornings for so many of the children, we don't actually have any good way of quantifying whether we're seeing a phase advance here and how big it is or how sensitive to the different light intensities that response is. But let's turn now to the samples collected in the evening and look at melatonin onset. So here we have average melatonin levels on the baseline night. We had an average baseline DILMO at 6:59 p.m., and that's occurring an average of 52 minutes before children's habitual bedtimes. And now here we have melatonin levels the evening after the morning light exposure. If you remember from the first study I presented on phase delay, response to evening light, when we average across all participants and intensities, we have an average delay of a 56-minute shift. But here you can see that the curves look very similar to one another. And in fact, averaged across participants and intensities, the average shift after the morning light exposure was not even in advance, but rather a 13-minute delay. So here we have evening phase shifts in hours for individual participants across each of the experimental light intensities. And the phase shifts ranged from a 52-minute delay to a 20-minute advance. Only 3 of the children exhibited any phase advance, and that was after light exposures of 20 lux, 120 lux, and 2000 lux, with the greatest advance of 20 minutes being in response to light at only 20 lux. Data from 2 additional children who completed the full protocol are missing from this slide because even though we were able to calculate a baseline DILMO for them, we were unable to calculate the phase shift because all melatonin samples collected on the final evening were already above our threshold of 4 picograms per milliliter. So one of those children received 500 lux and the other received 2000 lux. So those were our 2 brightest intensities. And so my first thought was, well, maybe they just advanced so much from this bright light exposure that morning that their melatonin onset occurs before we even started collecting our samples. That's why they were already above 4 picograms per milliliter when we started collecting. But here are the melatonin curves for each evening for those 2 children, the blue line being the baseline night and the orange line being the final night after the morning light exposure. And in both cases, it does look like melatonin onset occurs a bit earlier for the orange line compared to the blue line, but it doesn't appear that the evening rise in melatonin has occurred before we even started sampling on the night, and that's why they were all above 4 picograms per milliliter. So even though maybe they had small phase advances, it doesn't look as though these 2 children had significant advances in DILMO, even in response to these brighter intensities of 500 and 2000 lux. So given the high sensitivity to evening light that we observed in this age group, even at really low intensities, the lack of a consistent phase advance response is pretty surprising. As I showed, we were unfortunately only able to calculate the phase shift for one participant at 2000 lux, and they did show a phase advance, but it was only 12 minutes. At 500 lux, the two children we were able to measure phase shifts for had phase delays of 13 and 22 minutes. So one possibility is that it just takes a much brighter light to elicit a significant consistent circadian phase advance in this age group. But that again would be a little surprising given how little light intensity it took to elicit a strong phase delay. Our protocol consisted of one light exposure followed by an analysis of phase shift later that same evening. And so it's possible that the circadian system just needs more time to integrate that signal. And maybe we would have observed a clearer phase shift in DILMO if we assessed melatonin onset the following evening. That would have been a little tricky to get parents and children to remain in the dim light in this protocol for a whole extra day. Or it's also possible that several mornings of light exposure would be needed to elicit a phase advance response here. But I will say that there are studies with adults in which a morning light exposure was found to advance the timing of DILMO later that same day. So here in this first study, they had 1 hour exposure to green light at 350 lux between 7 and 8 a.m. And they found that it advanced melatonin onset that same evening by an average of 19 minutes as compared to baseline. In another study, there was a 3 hour exposure to light, a bright light of 6500 Kelvin at 3000 lux, and it resulted in an average phase advance of melatonin onset later that same evening of 42 minutes relative to placebo. And so 19 minutes and 42 minutes are not huge advances, but they still observe consistent advances in response to one light exposure that same morning. And again, this is in adults who we would expect to be less sensitive to the circadian effects of light exposure than children. Lastly, it's also possible that our light exposure was given at the wrong circadian time to elicit a strong phase advance response. We don't have a phase response curve to light in this age group, something that I would love to do in the future if we can figure out how to do a forced asynchrony with preschoolers. And we don't know the timing of T-min or the length of tau. And so it's a bit of a guess how much of a phase advance we would expect from a light exposure at the circadian time at which we gave it. But we can look at adolescent phase response curves that have previously been established to 80 minutes of 5000 lux. As far as I know, this is the youngest age group in which a PRC to light has been established. And in this figure, time on the x-axis is the start of the light exposure relative to an individual's baseline DILMO. So I've highlighted here in red the range of time that our light exposure started relevant to children's baseline DILMOs, with the red asterisk denoting the average timing. So if we assume that the PRC to light in young children would be similar to this in adolescents, which who knows, it would suggest that we weren't hitting at the peak of the phase advance response for sure, but that our average timing of the exposure still falls at around the 1 hour advance mark for bright light in teens. And we initially ran 3 participants in a pilot version of this protocol, where we gave the light exposure an hour earlier than the eventual full study, starting 1 hour before habitual wake time. So now we'd actually be here in the adolescent PRC, closer to the peak phase advance response to bright light. And here are the results for those 3 kids. We used only intensities of 100 lux or lower in that pilot, since we expected a really strong phase shift response based on our data from the evening light exposures. So we wanted to see a little bit of variability in the response and not just saturate them with the bright light. But what we actually observe is a 17-minute phase advance after exposure to 10 lux, and then phase delays again of 8 minutes and 24 minutes for 80 and 100 lux respectively. So again, given the high sensitivity that we observed to evening light, even at lower intensities, we would have expected to see some more consistent advances in the circadian windows in which we gave our light exposures for this age group. So overall, we're still trying to make sense of what we're seeing in the initial data from this study and what its implications might be. It's unfortunate that we were unable to quantify a phase shift in the timing of DILMOF, where maybe we would have seen more consistent advances. But I welcome any and all suggestions or discussion on all of this as we continue to think through these results. But to sum everything up, our findings in preschoolers indicate that young children are highly sensitive to light exposure before bedtime and suggest the lighting environment may play a crucial role in the development and maintenance of behavioral sleep problems through impacts on the circadian timing system. This points to evening light exposure as an important modifiable factor in children's environments that could lead to improvements in their sleep. Conversely, our preliminary data with morning light suggests that young children may be less sensitive to the phase advancing effects of light in the morning. But clearly, we have just begun to scratch the surface on this topic. A lot of questions remain, and much more research is needed to understand the development trajectory of the human circadian system and the role of the lighting environment in sleep and circadian health. So with that, I want to thank you all so much for your attention. Thank you to everyone involved in this research and our sources of funding. I'm happy to take any questions now with the time that's left, or please feel free to email me at any time at the address on the bottom of the slide. Thank you.